Hello there. My name is Alexia Stokes, and I am an undergraduate student from Dr. Midlars's lab. Today, I'm going to be doing a poster presentation about elucidating the etiology of core diseases through symbiont gene expression. Before I continue on, I have to go over some things in the background. Symbionts are dinoflagellate algae that play a critical role for host metabolism through photosynthesis, respiration, carbon translocation, and calcification. Based off our first figure of coral polyp holobionts, symbionts are found in the gastrodermis between the mesoglia and gastrovascular cavity of the polyp. Two diseases that we will focus on in this study are white plague and stony coral tissue loss, also known as Skittle D. The etiology of these diseases are largely unknown, and thus the purpose of this experiment is to find a plausible answer to where within the holobionts these pathogens are targeting. Samples used in this experiment were collected in the U.S. Virgin Islands and were fragmented and put into take systems, which they were then separated based off of control and treatment. We did RNA sequencing and used these sequences to perform differential expression analysis and principal component analysis. We also calculated the eigenvalues based on this data set. Now, taking a look at results, we have differential gene expression results, which help to determine statistically relevant changes in gene expression when comparing control samples to diseased ones. We used bar graphs to visualize this data set in which we compared gene expression of both Skittle D and white plague in response to treatment with one of the diseases. The numbers in the bar graph represent how many genes were differentially expressed. So let's take a look at figure B for this as an example. Here we have both the symbionts and its host species exposed to Skittle D and white plague. In the red, we have the symbiont response, in which when it is exposed to Skittle D, it shows differentially expressed genes. Meanwhile, when exposed to white plague, it shows none. The host species, represented by the purple and blue in this case, show differentially expressed genes when exposed to Skittle D and white plague. In figure E, it represents only symbiont responses to treatment. When exposed to Skittle D, 3 out of 4 symbionts that we are testing show differentially expressed genes, while when infected with white plague, none showed any reaction to it. Another thing that we have done is principal component analysis, which shows what variation is present in all the samples' genetic expression. We can use eigenvalues to determine what phenotype is driving these variations. The phenotypes we measured are disease status, treatment, and host species. Disease status represents the state of the coral holobiont based on whether it is healthy, exposed, or diseased. And we did this analysis based on data collected from clade C. Based on this, PCA of white plague infected symbionts showed variation only based on species host phenotype. Meanwhile, PCA of Skittle D showed variation based off of multiple factors such as species host phenotype, disease status, and treatment. With these results, we can come to several conclusions. One of these conclusions is that based off of PCA clade C treated with Skittle D, there's little to no correlation associated with treatment and gene counts when infected by this disease. Most of the variation came from host species. Meanwhile, PCA clade C treated with white plague showed that there was no correlation between host disease fate and treatment and gene counts when infected with white plague. All variation came from host species. In addition, whenever we look at DAGs, we can see that symbionts treated with Skittle D showed a reaction to the pathogen, while those treated with white plague showed no reaction. Meanwhile, when we look at host species across the board, all of them show differentially expressed genes when exposed to white plague and Skittle D. Our results support previous studies that Skittle D affects symbionts within holobionts. From previous histologies, we know that Skittle D affects the gastrodermis where symbionts are housed. This is consistent with our results that suggest Skittle D is a disease that affects both the host and the symbionts. We can also see, based on other studies, that white plague seems to be a disease that affects mainly and only the host species. As the end of my presentation, I would like to thank the Midlars Lab and our collaborators for their support on this experiment.